welcome to this screencast on Mendelian Genetics. So we're going to build on the idea of meiosis and really looking at how chromosomes and the characteristics and the genes that those chromosomes carry get transmitted from parent to offspring. So we're still on big idea 3, looking at storage, retrieval, transmittance of this DNA information and how this is essential to the continuity of life. So what we've done so far, just a reminder that we just finished up the cell cycle in mitosis, looking at how new cells are created asexually, identical offspring, and then meiosis, how are gametes formed? How are those sperm and those egg cells created? And what are those 3 very important ways that variation can come about? And then today we're going to look at just basic Mendelian genetics, kind of lay the groundwork for some later work that's a little bit more complicated, so non-Mendelian genetics, and then finally chromosome mapping, which is really cool. So what should you take away from this? What should you remember next year and beyond? Well, it's just the idea that the heritable information that we are talking about, the transmittance of this information, provides for the continuity of life. And later on in the year when we talk about evolution, we are certainly going to come back to this idea of the receiving and the transmittance of information. So the really important essential knowledge is called the chromosomal basis of inheritance, and this provides an understanding of how genes are transmitted from parent to offspring. So it's really, really important to remember as we go through this, and this is going to get complicated, and this is going to be a hard unit, but we need to remember that what we're talking about are chromosomes. So when we talk about all these genes and alleles and traits, they're carried on chromosomes from parent to offspring. And how do they get to the offspring? Through the gametes, okay, and through meiosis. So just keep that in mind. Now, I'm going to use a lot of vocabulary that I'm not necessarily going to define in this video. So please take a moment now and look through these terms. And if you do not remember what these terms mean, you need to pause your video and write what these terms mean before moving on. If you feel like you're okay, then go ahead and move on, but I'm not going to explicitly define these in this video. Okay, so the picture that I just showed before, this is Gregor Mendel. He's a very famous, he lived in the 1800s. He was actually a monk. And what did monks do back in those days? Well, they obviously transcribed Bibles and they prayed a lot, but a lot of what they did was they had to take care of themselves. And Mendel was actually a gardener because he had to garden the food and make the food for the other monks. And so he started to study these peas in his garden. And through his studies with these peas, he came up with 2 fundamental laws of inheritance. And as we go through the next couple weeks, everything we talk about is based on these 2 laws. So it's important to understand these fundamentals. And so the first one is called the law of segregation. And this really applies to monohybrid crosses. Monohybrid meaning 1 trait. And so what this says is that 2 alleles for a heritable character are going to separate during gamete formation, and they're going to end up in different gametes. Now this should make sense to us, especially since we just finished meiosis. So let's just do a very simple example. So we have a mom, okay, and so she's got 2 chromosomes, one from mom and one from dad. Well, one is carrying a big P and one is carrying a little p. Well, when she performs meiosis to produce her eggs, these alleles are actually going to go into different gametes. So one is going to have a big P and one is going to have a little p. And you know what? Same with dad. Dad has 2 chromosomes. He's got big P and little p. Law of segregation says that they are going to separate into 2 different sperm cells. So the gametes are not going to be identical because the mom did not have the identical alleles for P and neither did dad. So law of segregation simply says that these 2 guys are going to separate from each other. The second law is a little bit harder to understand. It applies to a dihybrid cross or more traits. So di meaning 2. So this says that each pair of alleles is going to segregate independently of the other pairs during gamete formation. So again, we're still talking about meiosis, and this term here should look familiar because we did talk about how during metaphase they can line up in any way on that metaphase plate. So now we're going to see what this looks like as far as traits. So if we look at this generation here, okay, don't worry about this for right now, but when we look over here, when we see the gametes being formed, so these create these gametes, and these guys create these gametes. So here are the sperm and here are the eggs over here. Big S can either go with big B or it can assort independently and it can go with little b. Likewise, little s can go with big B, or little s could go with little b. They're not connected, and this is obviously assuming that they're on different chromosomes. But the chromosomes themselves, the alleles themselves, are not connected, and they're going to sort or segregate independently of one another. Mendel, let's go back to Mendel and his study of peas. I want you to write down in your notes why he used peas, and I think that you can come up with at least 3 very good reasons as to why he used peas as his model organism. He tracked at least 7 different traits, and they were all discrete characteristics, meaning they were either or. So they were either purple flowers or they were white flowers. So here's an example of a purple flower. They were either green peas or they were yellow peas. And so that was easy for him to follow in his breeding program. You do need to understand the 3 generations that he was looking at. The parental generation P was always true breeding. So for example, it was big P, or if it was white, it was little p, little p. Now what he would do is he would cross the parental. So he would cross big P with little p, and he would get the F1, which was their offspring. And the F1 was always a hybrid in this way, obviously, because it would get the big P from one parent and the little p from the other parent. Then in order to get the F2, and this is very, very key, he would self-fertilize. So he would take his hybrid, big P, little p, and he would self-fertilize. Because it's a plant, you can do that. And that's how he would generate his F2 generation. So let's take a closer look at this. So this is looking at the probability of a monohybrid cross. You do need to know these generations, and you do need to know the ratios that come about in monohybrid, and then we'll do dihybrid next. So again, we're going to use this idea of purple crossing with white. So we've got the true breeding parents. They create the hybrid offspring, which is a heterozygote. And then we're going to self-fertilize with another F1, so a heterozygote crossed with a heterozygote. And then when we fill in our Punnett square, okay, hopefully you remember doing this from a few years ago, big P, little p. This is the genotype ratio. Now what about the phenotype ratio? The phenotype ratio is we have these 3 that are purple, and we have 1, this little guy here, that is white. So you always, when you are doing these classical genetics with P, F1, and F2, you will always see a 3-to-1 ratio in the F2 generation. You have to remember that, because when we look at some data, you're going to be able to use these numbers and these ratios in order to basically extrapolate to know what the parents looked like. Okay, I want you to do this example, so pause your video, write this down, and show me your work for this example. Now moving on to dihybrid, I do want you to know the common ratios that occur in a dihybrid cross, so that should say dihybrid there. So parental, the one that your book uses, we're just going to use the same one. So again, we've got the true breeding yellow round, and Mendel crossed it with a true breeding green wrinkled. So little y, little r, little r. You should be able to predict right now what the F1 looks like without even really thinking. It's a hybrid, so it's going to be heterozygous yellow and heterozygous round. Remember, what do you do with the F1? You self-fertilize, so you're going to cross it with another hybrid. And I'm not going to show you this whole big Punnett square, but I do want you to know the phenotype ratios that come about from this. And this is a 9:3 to 3:1 phenotype ratio, and you do have to know that. Again, you might be looking at some data sometime in the future where this ratio pops out to you, and you say, ooh, I recognize that. That's a dihybrid, and I can therefore predict that the parental generation were in fact true breeding. I want you to do this dihybrid example with me. There are some basic steps to follow in setting up a dihybrid cross. It's a little bit more complicated than the monohybrid cross. So we're going to take this cross as our example. The first step is to write out all possible gametes for each parent. So let's take a look at this guy here. So let's take a look at this guy here, and we're going to write out the possible egg gametes for this guy. So big Y can go with big R, or big Y can go with little r. Little y can go with big R, or little y can go with little r. So there are my possible eggs. And then I'm going to take this guy and I'm going to do my possible sperm. And it's going to look exactly the same. Okay, second step is to put these gametes across the side and across the top of a Punnett square. So I will do that, but then what I want you to show me is I want you to show me the completed Punnett square in your notes with genotype and phenotype ratios. Okay, and here's another dihybrid example. Pause your video, write this down, and I want to see another Punnett square in your notes showing me that you understand how to set up a dihybrid cross. So first, write out this cross. Okay, so make sure you understand what the notations are. Then write out the gametes, and then put it into your Punnett square. Okay, moving on to probability. I just went over Punnett squares, but really, as we get into more difficult genetics problems, you're not going to be using Punnett squares. Instead, you are going to be using probability to solve these. So hopefully you're a little bit more comfortable with math than you were a few years ago because we're going to be relying on some math. So the first law is the easy one. It's the law of multiplication. So you're simply multiplying the probability of one event by the probability of another event. The easiest way to think about this is with a coin toss. So let's just do a quick example. What are the chances that the coin will land on heads 2 times in a row? So what's the chance it's going to land on heads just once? Well, hopefully you understand that that's 50%, so that's half. Well, what's the chance that it's going to land a second time? Again, it's half. It's independent of what happened the first time. So what we do is we simply multiply these 2 events, and the chances that it's going to happen 2 times in a row is 1/4, or 25%. Let's apply this same thinking to a genetics problem. So what's the probability of getting little r in a big R, little r times big R, little r cross? So let's rewrite this cross. So what is the chances that you're going to get a little r from this parent? The chances that you're going to get a little r from this parent is 1/2. And you have another half a chance of getting the big R. What's the chance of getting a little r from this parent? Hopefully you understand, again, that it's half. So just like before, you multiply. So the answer is 25% chance of getting little r, little r. So that's the easy law of multiplication you use a lot. The law of addition is a little bit hard to understand when you use it unless you've done a lot of examples. So this is the probability that any one of 2 or more mutually exclusive events will occur. And this is calculated by adding together the individual probabilities that you got from the law of multiplication. So you're actually kind of using both in a lot of circumstances. Again, easiest way, let's do an example. So here's the cross that we're going to do our example with, and we're going to ask the question, what fraction of offspring will be predicted, because remember, we're just predicting things with probability, to exhibit the recessive phenotype for at least 2 of the 3 characters. So these are mutually exclusive events. They could be little p, little y, little y. They could be little y, little r, little r. But for at least 2 of those characters, what's the probability that they're going to be the recessive phenotype? So first step for doing this is we first have to write out all the events that fulfill this at least 2, right? So I went ahead and I did that for you. So these are all the possible offspring that are possible and that have 2 recessive characteristics. So this one's got the P and the Y, this one's got the P and the R, and you can go on and you can see how I did that. So now what we have to do is we have to use multiplication for each genotype, and then at the end, we're going to add all of these together. So I'm going to fill this in for you. So I'm going to fill the first one in for you, and then I'm just going to fill the rest in, but I'm not going to explain it. So what is the probability from this cross that we're going to get little p, little p? Okay, when we look at the p's, big P, little p cross with big P, little p, the probability of getting little p is 1/4. And if you need to do a Punnett square to show that to you, you can do that. What's the probability of getting little y from big Y, little y cross with little y? That is 1/2. What's the probability of getting big R, little r from big R, little r crossed with little r? That is 1/2. Once I got all my individual rule of multiplications, then I added all these together, and the answer is that 7/16 of the offspring are going to display at least 2 of the recessive characteristics. The best way to learn the rules of probability and to learn genetics is practice, 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 and time. So I've given you 5 practice problems to do to get you started. You're also going to have a whole packet to do. I know you're so excited, but let's start with these 5 and we'll go over these in class. When you do these, I want you to use the rule of probability and not Punnett squares. And the fifth one is right here. See you in class.